So it's October 2nd, we've got our strand of DNA, it's been replicated, the cell's copied, all that has happened, and now we have to find a way to make a one-sided copy of this molecule, just a portion, to get it out of the nucleus. So I'm going to go ahead and I'm going to get rid of that, because we're really only interested in one half of the DNA at this point. We copy one half at a time. And what we're going to do is called transcribing. This process is called transcription. Transcription. Do you know what it means to transcribe something? Have you ever transcribed something? I bet you have. Have you ever copied something down? Have you ever copied a friend's notes? Have you ever copied a recipe? Have you ever copied directions for something? That is transcribing. A scribe is someone who writes something. A script is something that has been written out. To transcribe is to copy over. Okay? Same language. We're not changing the language here, but we are copying something over in some way. So we take half of this DNA and we do this process called transcription, and what we get is a molecule called RNA. And I'm going to change the backbone color. Well, no, I guess I'm not. Okay, so I'm going to show the RNA right here. Now, RNA is ribonucleic acid. So deoxyribonucleic acid is DNA. Now we're dealing with ribonucleic acid. It's real similar. It's got a sugar phosphate backbone. It's a slightly different sugar. That's why it's got a slightly different name. And it's only one-sided. But it's still made up of 4 nitrogenous bases. There's just one little catch. There's no thiamine in RNA. You know this. There's no thiamine. Yeah, there's something called uracil. And uracil replaces, oops, I had thiamine as red, huh? Thiamine. Uracil replaces thiamine. There, it looks like a little Italian flag or Christmas decorations of some sort. I don't know what it looks like. So any place that we would normally put a T in as the complement, we're going to put a U in now. Beyond that, though, it all matches up. It's all, well, I can't do my color codes here. It all matches up. So cytosine still pairs to guanine, guanine still pairs to cytosine, thymine still pairs to adenine. There's adenine there. Cytosine again pairs to guanine, but every time that we hit adenine, the complement to adenine in RNA is what? What is it? Uracil. So we have U's in there instead. Thymine pairs to adenine, guanine pairs to cytosine. So now we have this half molecule, this one-sided piece, and I'll get rid of this. And that's small enough to come out of the nucleus. That's small enough to leave through those nuclear pores and go to the place where proteins are built. Where are the proteins built? Ribosomes. They're built out in the ribosomes, little Pac-Man looking things out in the cytoplasm. So now that moves away from the DNA and heads out into the cytoplasm. I'm going to switch the side that the backbone is on just to make this easier. And once it gets to the ribosome, basically trucks are going to deliver goods. That's the easiest way to think about it. And the goods that those trucks are delivering are what are called amino acids. So if we imagine a little truck, and that little truck is going to line up its license plate to these 3 bases in the RNA. And these 3 bases are actually what code for the amino acid that's being brought by the little truck, and it's called a codon. Okay. Now, the anticodon is sort of like the license plate on the truck, and it lines up to that codon, so I'll label that. And it sort of provides a check. Yes, is this the right amino acid? Does the license plate match the codon? Yes? Okay, we're good. Well, at that point, then, the anticodon goes away. And that amino acid is sitting there just waiting for something to get joined to it. Well, we have another anticodon that comes along to match the next codon. And let's see. That would be U, what? C. And that's going to have some other amino acid on there. We'll make this one bright yellow. Some other amino acid. The anticodon sort of checks the license plate. Do they match? Yes? Okay. So that's another amino acid. Those two things can be joined, and those two things are joined with a very special kind of bond. This is called a peptide bond. Okay. And we could keep going all day, adding amino acids. I'm going to get rid of my little anticodon here. I'm going to get rid of my label. Well, I won't get rid of that just yet. Maybe I could move my label. Maybe I can't. Get rid of that. So those are amino acids. This peptide bond is what holds them together. And we just keep adding amino acids. We could keep adding amino acids all day long. And they keep getting joined by peptide bonds, which is why proteins are sometimes called polypeptides. What does poly mean? One, two, or many? Many. So a polypeptide is something that is held together by many peptide bonds. These amino acids come from the food that you eat. Your body's able to synthesize some of them your body can't synthesize, so you get them from your food. You get them from basically the protein and other things. And that's kind of it in a nutshell. We take the DNA, we transcribe it into, and this is actually called mRNA because it's the messenger that carries the recipe from the living room, from the bulky computer into the kitchen, carries the message from the nucleus out to the ribosome where the stuff is being built. And then those little anticodons, I should have mentioned, those little anticodons, and I'll just make one real quick here. Yeah, what am I doing? Jeez Louise, Moser. A, U, G. These little anticodons are also a form, they are also a form of RNA. They drive the truck. So these are actually tRNA. Can you guess what T stands for? It's not truck. Transport. It's transport RNA. It transports the amino acids over to the ribosome so that they can be hooked together to form proteins. This is not a complicated process. Well, it is. At the level that we're looking at it, it's really not. We're looking at it at the simplest, most basic level. And remember, we're looking at this right now in eukaryotes. Just because it's a good place to start We're going to sort of get this down and make sure you know how it works in eukaryotes. Then we're going to talk about how the process is different in prokaryotes. Because why are we doing this again? Because we want to understand how a bacteria can be resistant to an antibiotic. And a lot of that has to do with how, honestly, the protein synthesis process. A lot of antibiotics attack this process in bacteria. If you can stop a bacteria from producing proteins, you can stop it from building its protein cell wall. And if it has a weak cell wall or a cell wall that isn't fully built, then it's easier to kill. What I have for you next is a little worksheet. We're going to walk through, well, actually, we should walk through the Amoeba Sisters together. Okay, so I forgot to hit record. So we did the portion of the mutation, and we did the complement of mRNA, and now we're going to look at our protein, our amino acid chart, to figure out what amino acids we've just coded for. So if you look at this wheel, this is the easiest one by far to use, so I'm starting you off with this one. The one that they typically put on the Ohio Air test is more complicated, so I want you to kind of understand the concept before you go to the harder chart. On this one, we're starting at the inside wheel, and whatever your first base in the codon is, that's what you're looking at. So in this first codon, what's our first base? C. So we're starting in this quadrant. Then we look at the next wheel out, and we go to our second base, which is also C. I'm trying to change colors every time just so you can follow this more easily. And then we go to our third base on the outside wheel. And in this case, no matter what that last base is, they all code for the same amino acid. So going through U takes us to, what's the base? Or what's the amino acid, rather, I'm sorry. It's pronounced proline. Proline? Not actually sure, I guess. So the first amino acid, I've always said proline, is proline. That didn't change. Well, we would expect it not to have. Our mutation wasn't in the first codon. Do you need help? Okay. Good? Okay. Well, let's go to our second codon. Our second codon starts with a G, so we're going to start here. Then we have a U, so we're going to go here. And again, it doesn't matter what the third base is. What amino acid are we coding for? Valine. Is that the same as what was coded for in the original strand? No. So that was a mutation that actually changed the sequence. It actually changed the sequence of amino acids. Now, our third one, GAG, our last codon, we start with G, we go to A, and then it does matter what our last base is. A and G get us glutamic acid. Now we want to contrast that with a Frameshift. So this only changed one amino acid in that protein. Now, that's enough. This is the single mutation that actually causes a disease called sickle cell anemia. We'll talk about sickle cell anemia when we do some heredity stuff a little bit later in the year. It's a disease, it's not always fatal, but it can be. It predominantly affects African Americans, and there's one other sub-ethnic group that's prone to it, and I cannot remember who. Like 1 in 12 African Americans carry this mutation, so it's a big deal. And it causes a lot of pain because the hemoglobin protein isn't formed correctly, and you get what are called sickle-shaped blood cells, and they tend to clog up in the smaller blood vessels in the body, and so you can have you can have loss of oxygen to the extremities. People with sickle cell are really prone to infection throughout their lives, so it's rough. All right, so now we're going to look at what happens, and that's just a substitution. That's a single base shift. But now let's look at what happens when we have something that causes a frame shift. So, an insertion or deletion can cause a frame shift. There's our normal strand. What I'm going to have you now use your wheel. Well, we'll do the mRNA together. So for GCA, what's the mRNA codon? G gets a C, C gets a G, A gets a, be careful, U. A gets a U, mRNA. T gets a, C gets a, G. Oh, and I just wrote, G gets a C. How about we try that, Moser? Jeez Louise. I'm just trying to keep you on your toes, that's all. It's all for you. You're welcome. Anytime. Okay, and then CAC, what would the codon be for that, the mRNA? G, U, G. Gug. These make such nice three-letter words. Okay, using the wheel, I want you to figure out what the amino acids are. So a lot of people get to here and they go, well, wait a second, I can make mRNA of UG, but then I don't know how to read that on the codon chart, and neither does the cell. Which means the whole mechanism is disrupted. And you can't code an amino acid with only 2 bases. So that spot would be left blank. There's basically no amino acid added to the sequence. It's not, we haven't talked yet about start and stop codons. It's not an official stop, but yet the cell the mechanism kind of breaks down and the protein gets cut short. Okay. I'll let you answer those 2 on your own. But I want to look at the worksheet with you. The first thing that this worksheet is asking you to do is to just get really good at being fluent at going back and forth between transcribing from DNA to mRNA. Remember, this is transcription. We're not changing the language. There's another vocabulary word we haven't introduced yet. But we're just looking to see if we can figure out, well, if this is the mRNA, what would the DNA have been? So what DNA bases would get you an AA, G, T, T, C? Okay, that's easy enough. And then we want to know basically what the license plate on the truck, what the tRNA anticodon would be that would match up with this AAG. What would it be? U, U, C. Then you need to go to the chart and find the amino acids. This is important. This is big. Put a red star on it, put glitter on it, whatever. The amino acid is coded for by the codon, not the anticodon. The tRNA is just the check. It's just like matching the license plate that delivered the thing. So the amino acid comes from that anticodon, or from that codon. Geez Louise, Moser. Comes from the codon. It does not come from the tRNA. Okay, I'm going to let you work on that. Okay, so we said when we go from DNA to mRNA, that's a transcription. We're not changing languages, we're just transcribing it. But when we go from mRNA to a protein, that's a totally different language. We're going from a nucleic acid, of course with the help of this other nucleic acid, and we're going to a whole different thing. And that whole different thing, that whole different language means we're actually translating. We're translating the nucleic acid into a protein. a series of amino acids linked with peptide bonds.